Hi, this is Dr. Jabali, and we're going to do the video on topic 1.6, multiple choice. Remember, all these questions are coming from previous IB exams, and we're going to learn how to tackle them and understand the IB language. Remember that whenever you're going to start doing these multiple choice, make sure to read the question twice carefully. Some people need 3 times, as well as the options A, B, C, and D. Read it very careful because sometimes IB can trick with the answers. Sometimes you're going to reach yourself seeing 2 questions that are very close in answer. It's always the one that is more specific is the one to go for. So with that, let's start here. It says a tissue sample was examined under microscope, and in order for it to determine the mitotic index. Now, if you have studied the 1.6, you understand the mitotic index is a tool to see if there is a lot of cell division. And if there is a lot of cell division, that means there is cancer. So the number of cells in each stage of the cell cycle was determined. That means we want to see how many there is in prophase, anaphase, telophase, so forth. And you can see the data was put in the table. So here we have the number of cells, and here we have the stages of life. In total, there was 160 cells. So that means, what's the equation for mitotic index? So the mitotic index, and this you should know it, you should do it by heart. This is basically the total cell, that means total cells there is in the tissue that they sample they took, and here how many, the number of cells dividing. So we already found out that the total is 160. So we already know it's 160. Now we need to find how many out of that 160 are actually going through cell division. Interphase, there's no cell division going on. So it's basically during this period of here, which we have 20 plus 10, 30, plus another 10 here, 40. So you can see that 40 out of those 160 is going through the process. Now, you want to find out what the mitotic index is. So you cancel this and you cancel that, and then you just divide 4 by 16. And that probably will be around, if we do the calculations, 40, that's 3 will give 6 stuff. And most likely that will give us 0.25. Here's the bad news. This is basically a paper 1. So that means you're not allowed to use a calculator. So you really have to be good in doing the divisions. If you can't do this, there's always ways to simplify. We know that 4 divided by 4 is 1, and you divide it here also by 4, and that's 4. So you can see 1 out of 4, and that is half or quarter, right? So this is what the answer is. So they're always going to give you some kind of calculations when it comes to paper 1 that it's easy to do in the head, okay? Next, it says here, when does DNA replication occur? Now you have the cell cycle, just as a refresher. We have G1 as G2, and then we enter mitosis, and then mitosis cytokinesis, right? And then back to G1. This is usually growth, this is DNA replication, this is growth again, and then mitosis is division, and cytokinesis, you divide the cytoplasm into 2. So, like a quick refresher. Now, we're looking at the question. It says, when does DNA replication occur? So that means DNA replication means I am making a copy of DNA, and that happens on the S. So you can see that the only answer we have here is A, because that's the S phase of the interphase, and that's the answer. Now let's go to the next question. This question here shows the graph shows a mitotic index in the roots of lentils. Lentils is a type of plant at different distances from the end of the root, right? So we have here the mitotic index, right? This is the division after you did the number of cells dividing over the total amount, right, of cells. So this is the different values from 0 to 14. Here is the distance from the end of the root. When we say the distance from the end of the root, we're talking about, let's say, here is the plant, and you're looking from here, distance to the root of the plant. What is the distance it has from here to here? So let's work at this. Now, what can be deduced? Deduced, that means what's your educated guess about this graph, okay? Now, before we get into the ABCD, first let's look at the graph. Right, we can see as the distance is further away, the mitotic index is low. But the distance from the end of the root, when it's small, there is a bigger number of mitotic indexed. What does that mean? Usually mitotic indexed, if it has a large number, it means that there is a lot of cell division. So as I recap here, you can even write this. A large number of mitotic indexed, it automatically means lots of cell division. That's what it means. So let's erase this. And vice versa. So if it's a small number of mitotic indexed, it means little cell division. So from this graph, you can see that here there's a lot of cell division. And here you can see there is low. Most likely, think about it, the distance here. Here it's because it's still small, so it's growing. But here it's already basically 2 millimeters, which that means it hasn't grown enough. So it doesn't have to keep making more cells to grow. So that's what our assumption is, right? Because if you want to keep growing, you have to do more cell division. And if you don't want to grow anymore, you stop the cell division. So with that in mind, let's look now at the different options. A says, as the distance from the end of the roots increases, more cells are undergoing mitosis. So we already just did this, right? As the distance increases, we said that there is less. So A is not it. Next, it says, at 0.5, which is this right here, from the end of the root, most of the cells are in prophase. Is that possible? Yes, it is possible because prophase is part of the cell division, and we see that there is a cell division, but don't answer quickly. You still have to look at the other two. Here it says there are fewer cells observed at 1.5 than at 0.5. This has nothing to do with because if you want to make a graph, you have to have a control. That means, what do you mean by control? So it's talking about 1.5. When they say fewer cells observed, you're basically saying that, let's say mitotic index, let's erase this to give you what I'm talking about. When you want to graph something, right, the first thing you have to do is go through the process of experiment. So if you want to look at the 100 cells, then over here you have to look at the 100 cells, over here you have to look at the 100 cells and 100 cells and 100 cells, because it has to be controlled. Because then the graph does not make sense and it's not reliable. If you're saying that around here we look at the 100 cells and here we look at 50 cells, of course the mitotic index is going to come different. And if you here is only 10 cells that you're looking at, it doesn't make sense. Because again, going back to the equation of mitotic index cells, it's the cells that are going through the cell division over the total of cells there is. So to keep it control, all of them should have a total amount. That's if it's 100, then all these different distance you have to count as a total of 100. So if there's a total of 100, that means you have to observe 100. So you can't say a few of cells will be observed at 1.5 compared to this, because then you do not control your experiment. So this is out of the way, these two, right? And then the last one is, as the distance of the end of the root increases, so it's increasing, the percentage of cells in interface increases. So here we're saying that the cells, right at this point in the interface, increases. Is this true? It is possibly true because what? The mitotic index is less, right? But then we are stuck at 2. These two right here are very much like, uh-oh. But then this is where I told you sometimes we have two sentences that are very close, but let's erase this here. Let's give you a little bit more focus so you can see how you can easily decide by the two. Some people already picked their idea, but for those who didn't, so they have this one and this one. It is true, like here it says increases, right? So once you get here, the percentage of cells in the interface increase. You're saying that there is more cells in interface, right? However, this is not talking about after you produce the division, right? It is true that if by that point you already had produced more cells, but this is we're talking about cutting a tree. Let's say I'm cutting the tree at the same time at this level of root, this level of root, and this level of root. All these will cut at the same moment. It's not like you cut this one and then you waited for grow to become this and then you waited this to grow. Because if it was, then D would have been the right answer. But here is just saying I cut these roots, right, at exactly the same time. So they should have 100 cells exactly at the same time. And as I observe here, I can see there's a lot more division. I observe here, there's a lot more division. Here, there's a lot of division. Here, you start to reduce the amount of cells that are division, and there's more interphase. But we're not saying that the percentage of cells in interphase has been increased. This is not showing basically that you have made more cells and more of these cells are interphase. So the right answer in this case would have been this one, because at the end, most of the cells are in prophase, in which they're about to go to mitosis, and eventually they're about to grow and become bigger cells. That's why it's still at the 0.5 centimeters, because it's still a baby and the root is still growing. So most likely they still have a lot to go to become as big as 2.0. So between the B and the D, the answer obviously will be B, because again, the bottom one, the D one, is telling you that you don't have any more 100, right? You're basically saying that there's like 200 and there's 100 more cells, and these cells are in the interface, and that's not what it's trying to say. Very tricky one. But again, the answer is B. So let's move on to the next question. This is a process in the shape. Now, we already went through these questions in the previous videos, but remember that the cell cytokinesis, for example, in animals or plant cells, they usually will look, this is what the animal will look like. In a plant cell, cytokinesis will usually look something like this. And then in the bacteria, it will usually look like this. So you can see the animal, the plant, and the bacteria. Now this resembles a lot more the bacteria because it's more of a rod shape, right? It's a bacteria that is dividing into two. So since it's a bacteria, we know the bacteria go through binary fission, while these two, they're going through mitosis. So let's look at the options we have here. First, binary fission in prokaryotic cells. That could be the answer because, again, bacteria is prokaryotic. Telophase 2 in eukaryotic cells. It is not because, again, if it was an animal, it would have been, the shape would be like this, more like a circle. So it can't be that. It's the endomitosis in prokaryotic cells. First, prokaryotic cells do not do mitosis, so that already canceled. It's binary fission. And again, cytokinesis of eukaryotic cells. It could be cytokinesis, but cytokinesis also means that they have to look like this shape and not like a rod shape. So the right answer here is A, because of the shape. It really shows that it's a rod and it's a bacteria. Now moving to the next question. We have here many times in IB, they will put micrographs showing maybe the prophase, metaphase, anaphase, or telophase of mitosis. So you really have to be going through these and make sure you study and see them a lot. So here it says there's a mitotic phase labeled X, which is this. It's a micrograph, and this is of an onion, of a root tip. Now, they want to know which stage is this one right here. So if you look at this stage, you can see that most of these chromosomes are in the middle. They're right in the middle of the cell. So middle of the cell is usually means, if you remember metaphase, it has the centrioles, and then you have the spindles going like this. And then you have the visible chromosomes line up in the middle like that. And that's exactly what this guy looks like. You can see it right here, that they are lined up in the middle. So the right answer for this one would be B. This is an easy one. So make sure, if you're still not comfortable with this, go ahead, look at BioNinja, look at your book, or look on Google Photos. Search micrograph pictures of mitosis, prophase, metaphase, and keep looking at many of them so you can get very familiar with these drawings. Now, this next question. When during the cell cycle does DNA replication takes place? Now, just as a recap here, right? This whole process here, let me highlight it here. This whole area here, all of this, it's even wrote interphase. All of this is interphase. And then this yellow part here is mitosis. Now, look at the arrows in which ways the arrows are going. The trick is that it's going in this direction. So once mitosis ends, what's here? It comes cytokinesis and then G1, right? And then after G1 we get S, and after S we get G2, and then G2 goes this. So it's a trick, right? Because usually when we study in drawings, we usually do the opposite. We write G1 goes to S, and then S goes to G2, G2 goes to mitosis, mitosis go back to G1. We usually draw in this direction, right? But here IB, like IB, always love to trick students. They did the opposite with the arrows. They flipped it. This is a way of tricking you. So we know that the S stage is where the DNA replication is happening. So the answer should be right here, the circle, C. That's the answer. So remember, make sure not to quickly go into the answers, analyze the pictures and drawings, because IB can flip things and trick it for you. So we get into the next question. So it's a graph. It shows the survival probability of current smokers. So this is relation to current smokers, okay? And for those who never smoked, among women of age of 30 to 8 years old age. So we have their ages and the X, and we're talking probability of them surviving and the 30 years of age. So what is the percentage of them surviving at above the 30 years of age? So here it means that they can 100% survive. Here it means they don't survive, 0% of survival. Here we're talking about 30 years old to an 80 years old, okay? So if you look at more closely, this is saying that as you get older, your chances of surviving cancer is dropping if you're a current smoker. Also here, it shows that as you get older, your chances of surviving cancer is also dropping. But between these two, right, between these two, you can see that the current smoker is dropping faster. So that means you have a higher chances of getting cancer if you're a current smoker. So this was just analyzing the graph. Now let's go to the possible options to choose. So what can be deduced? That means after analyzing the graph, what can do? Before I go out of this, let's just understand that it's extremely important that first look at the graph, really analyze the graph. And when you analyze the graph, look what's in the X and look what's the Y. You gotta see if this one here is increasing, will this one be increasing or not? If this one is decreasing, what is happening? So it's always try to find the relationship. If they're both increasing or they're both decreasing together, or one is increasing while the other one is not, or the other one is going up, stuff. So try to find which of these four relationship is the graph, because that's a way to start to analyze. In this case, if you look at this both as you can see here, as the probability of them having a chance to survive decreases as you increase age. So this is basically here. See, as the probability decrease, the chances of cancer, as the age increases, right? Or it can be this one here. As the age increases, the probability of surviving drops. So it's an inverse relationship. Try to find this about the graph before you even get to the options. So now we did this, let's look at the options. We have here A. There is a correlation between smoking and cancer. Now, this is in your book, and this is where it says, yes, there is a positive correlation between smoking and cancer. It means as one increase, the other one increase. But is this enough answer to tell you? No, because it only says there is a relationship, but it didn't tell you anything else. Do I already give this as an answer? No. You got to be very careful. You might have another answer that is more specific. So let's look. B. Smoking reduces life expectancy. Well, here it's talking about survival, right? So life expectancy means how long can you live? So the smoker here did actually have reduced more chances of survival compared to a non-smoker. So guess what? This one can also be. This one is very generalized, but it can also be. So these two can be. Now, smoking causes cancer. nothing here said about cancer. And just because smoking increases and cancer increases at the same pace or usually together, it does not mean that one causes the other. So they can correlate, but doesn't mean they're causing each other. So that's out. Now, the next one is 70% of the smokers survive to 80 years old. 70% of the smokers. Look at the 80 years old. Not even close. You can see that it's really low, as in 40%. There's no way. So this is out. So now you're stuck with these two. Now, here's where I tell you, be more picky. For sure, smoking reduces life expectancy. That's proven. This one says there's a correlation between smoking and cancer. But cancer was not mentioned here. Here it was only mentioned the survival, right? The survival. It didn't say what it died of. So you already, we studied that smoking and cancer goes together. No, my friend. Just because you study doesn't mean that you graph, you're going to put that. So this A is very generalized. The only thing you got to do is you got to focus on the answer that focus on the graph. And this one is the only one that really focus because it's talk about being able to survive according to smoking. It's not talking about cancer. So the answer is B. Now let's look at this one. This is a diagram that it shows the concentration of forced cycling during cell cycle. If you do remember, the cell cycle is controlled by cyclins, right? Oops, let me erase this again. I'm right here. By cycling. And it's basically based on four types of cycling, which is DABs. DAB right here. DEAB. And they are the ones regulate when you go from one stage to the next. Now, here is asking you which curve represents, right, the cycling that promotes the assembly of the mitotic spindle. So this was basically saying it's the cycling. If you remember from mitosis, you are making spindle. So this must be something related to mitosis. So it's the cycling that pushes mitosis to go forward. So when you try to look at this, we have four options. Here are the four options. It could be A, it could be B, it could be C, or it could be D. It doesn't even ask if it's the cycling D, E, A, or B. You just look at the lines. But before I even get to these lines, let's learn a bit cycling again for those who don't know. So for example, if you are in a situation when you want to go from mitosis to G1, right? When you wanted this, you need to have a lot of cyclin D. When you have a lot of cyclin D, then you go to G1. If you want to go from G1 to S, you have to have a lot of E. When you want to go from S to G2, you need to have a lot of A. And when you want to go from G2 to M, mitosis, you need to have a lot of B, right? So what do you mean? Because once you have a lot of a cyclin D, for instance, you activate CDK, which is an enzyme. And this enzyme pushes you to go, for example, from mitosis towards G1. But that's only when you have a large amount of cyclin. And the same thing when you cannot go from G1 to S unless you have a large amount of cyclin E. You cannot go from the S to G2 unless you have a large amount of cyclin A, and you cannot go from G2 to M unless you have a larger B. So here you can see G1, you're increasing the amount of B until a moment that you increase, when you're going from G1 to S, right? Because you're going from G1 to S, what do you have to increase? You have to increase a lot of E. Once you have a lot of E, boom, you have entered phase, S phase, and then they don't need any more of the E. From here, when you're going basically from the S phase, to go to G2, what do you need from S to G2A? So right about here, you have to have a lot of A. Once you have A, you enter G2. If you want to go G2 to mitosis, you have to increase what? A lot of B. And when you have a lot of B, boom, you enter mitosis. And this is how it goes. So basically, in this case, what is that promotes mitosis for you to enter? It's D. Let me erase all this so you can see. So which cycling do we want? We want cycling A? No, because this is not going to be working. If you notice this is cycling D. You don't need that, right? Because you're going from mitosis back to G1. We're talking about how do we get to mitosis. So that means how do we get from G2 to this? You obviously will need this guy to be high in concentration, so the answer is D. Okay, if you still don't get it, let me just put here the cycling so you can understand what we're talking about. So from G1 to S, if you want to go from G1 to S, you need E. If you want to go from S to G2, you need A. If you go from G2 to M, you need B. And to go from mitosis back to G1, we're at D. And that's how it works, DAB. But we're talking about how can we get here, because it says how you get to this. So we need to put D. All right, the next question here. Which diagram represents process used in asexual? When I say A here, sexual, we're talking about mitosis. If I only said sexual, then it would be meiosis. So here we have a cell that is splitting into two and sending its genetic material. Here we also have a cell that is splitting into two and sending its material. But what's the difference between these two? If you really look at closely, in this case, if it's mitosis, the cells, if this is dad or mom, whatever it is, the parent cell, what does the babies have to do? They have to look exactly identical, right? They have to look identical to the parent, which is this one. Does these two look identical to the parent? No, it doesn't. So even though it makes sense that it's mitosis, it's divided into two cells, daughter cells, but they're not identical. Look, it's just a T, a small t. This is identical. What about this one? Well, it's about a cell splitting into two. This is not. These two are trying to join to make one. This is an example of a sperm and an egg joining to form a zygote. So this is sexual. So if we look at the answers here, now let me erase this here. You can see from this drawing, I think 2 is more this. So is it 1? Is it A? No, because it's not this one. Is it B? 1 and 2? No, because this is also canceled, so this is canceled. Is it only 2? Most likely. Is it 3? No. So the answer is C. So look very carefully at what's inside of the cell. Like I told you, this looks a lot like mitosis because it's making two cells. But what kills is because it does not look like the dad or the previous parents. This one does. All right. Moving on to the next question. Here is an image, shows cells in different stages of mitosis. Remember, keep studying micrographs of mitosis, get strong on it. Now it's asking you to look at this picture and tell which one of them is actually telophase. Telophase is in the end of it. So when it's telophase, you start freaking out, right? But we have this one, this one, this one, and this one. So just look out of these 4. Now, out of these 4, which ones it's easy to figure out? This one is easy to figure out because it's still looking like a cell like this, and it's still looking like it has some dark areas. So this for sure is not telophase. Why? Because telophase, usually it would look like it's a dark side here and it's a dark side here. That's a telophase. So this is not it. This one, B, this is where students get tricky because B and D look very alike each other. So I will keep them to the last 2. Then I look at C. See, they're all in the middle. So they're most likely to be like metaphase when they're all in the middle right here. So C is out of the way. So then you are stuck, right? Here, let me see. You are stuck between B and D. Is it this one or is it this one? They both look like it's separating like this. But if you have to choose between the 2, the more they are distant from each other, because in anaphase, right, you already see them kind of separating like this, and you already have them separating like this, but they still be close to each other. So this is still close to each other. This is more further away. So looking from this, the answer would have been, again, D, because they're more farther apart from each other. All right, moving on. Now we have, how does mitosis produce two genetic identical nuclei? So we have here mitosis. If, let's say, A, when he's going to split into two, both of his daughters right here needs to be A. So the question is how? How does mitosis does that? How does it produce two genetically identical nuclei? So let's look at the options that we have here. First, A, by separation of homologous structure. See, homologous structure is part of meiosis. This is where you're doing crossover, so it has nothing to do with mitosis. As soon as you see the word homologous structures, remember, this has to do with meiosis, which I'll be explaining on topic 3.3. Next, the next one says, by separation of the sister chromatids. That's possible, and we're going to keep this in mind, so I'm going to put here as a question mark. The next one, by the division of the cytoplasm into two equal cells. This is also inconsidered, but I'm going to show you why this might not even be the answer. But I'm just going to say, okay, let's keep this out. So yeah, it's true. They can make two equal cells. I'm going to show you what's the trick. And then, of course, D, by division of the nuclear membrane into two equal parts. We never divide the membrane into two parts. The nuclear membrane disappears and then it reappears. So let me show you how we're going to debug, because I know these are three things that students might get confused, right? The first one is easy, so take it off. But let's look at D first, division of nuclear membrane. If you remember in prophase, the nuclear membrane disappears and the chromosomes become visible. That's what happens in prophase. This is trying to say that's not that. They're basically saying that in prophase, you made two nucleus, equal parts. And you're going to have chromosome here and you have chromosomes here. Is this what you do in prophase? No, the nuclear membrane disappears and then it only appears back in telophase. So this is out of the question. D is out. Let's debug the next one, C. C here says a division of cytoplasm into two cells. If you remember telophase, right? It looks like this shape, and you have the nuclear envelopes reappear, and you have DNA becomes uncondensed and visible. And telophase ends here. That means mitosis ends here. Did you actually split this? No. What's the next stage after mitosis? Cytokinesis. And cytokinesis, it basically means that you are pushing cytoplasm until you split the cytoplasm. So when you split the cytoplasm, voila, you have two pieces of new cells. So C is not part of mitosis. C is part of cytokinesis. So that's out, but some kids fall in the trick. So B is the right answer. And why is it B the right answer? Let me draw it here for you. This is part of the anaphase. If you go and you draw anaphase right here, you have the centrioles and you have the spindles coming out like this, and you have spindle coming out like this, and the sister has been split. Each sister goes to one side, right? Because the chromosome is to be like this. And this one is a chromatid, and this is also another chromatid, and they're both a copy of each other. So when we call them together, sister chromatids. In anaphase, what do? You split them. Each one goes to each side. And so this does make sure that each copy goes each way. You see one of the copy goes this way, the other one goes here, another copy of this one goes here, this copy goes here, and so forth. So you can see it that you're splitting and making sure that the two eventually new cells will get the equal parts of the copy to themselves. So the answer here is B. All right, let's look at this question right now. It says students examined micrographs and counted the cells in different stages of mitosis. So you already see that they're talking about counting cells, and you can see it's in different stages of mitosis. That means prophase, metaphase, anaphase. And they also checked to see cells with no visible chromosomes. When they say no visible chromosomes, they're talking about interphase. That means the time where there is no mitosis. It's very important that you remember, only during mitosis can you see the chromosomes. If you cannot see the chromosomes, then that means you're in G1, S, or G2, or basically during the interphase. So here are the results, right? You have prophase, metaphase, anaphase, telophase, and interphase. However, I did not give you the total. We have to have the total. So in total, how many cells did they look at? 10, 15, 20. So there is in total 50 cells. What is the mitotic index? If you remember that mitotic index is the number of cells in division, right? They're being divided over the total amount of cells. We already figured out that the total amount is 50. Now count how many going through division. We do not use interphase because interphase is a stage that there is no division happening. So we're only counting these guys. So we have 10 plus 5 plus 5, that's 20. So we can see 20 out of the 50 is the amount of what we consider to be the mitotic index. Now we have to do division. Remember, you cannot use calculator in paper 1, so you're going to have to use from your head. Now 20 divided by 50 is the same thing as 2 divided by 5, right? If you cancel. And in this case, we know that this is about 0.4. Because again, it's basically 20 divided by 5, which is 4, but it's because it's 2 out of 5, it's 1.4. So the answer is B. Sorry, but if you're not good in math, buddy, you need to start using that brain and start working on division numbers because, again, paper 1 won't allow calculators. So we're going to the next one. Next question here is talking about, which is the last question for this 1.6. It says how many chromosomes, so we're talking about chromosomes here, are there in a cell during the anaphase of mitosis. So recapping, what does anaphase looks like? You have here anaphase, you have the centrioles, and you have the spindles coming out. That's anaphase. Now they're asking how many chromosomes there is, right? One sister here, but we need to know. Now, here comes the next thing, information. It says if the diploid number of the cells is 20. So what does that mean? Diploid is basically 2n. What does 2n means? The total amount. So for example, look at us humans, for example, our diploid for humans, right, is 46. So for let's say for a haploid, that means half of that will be a 23. That's for humans. Okay? Now, whenever you're going through mitosis, you cannot put just 46 because there has to be 46 coming this way and there has to be 46 coming that way. So when you start mitosis, right, let's say during prophase, how many do you actually have? 92. And metaphase here, what do you actually have? 92. And then here you have 92 because you got to split 46, 46. So when you get to telophase, what are you going to have on each side? Right? A 46 and a 46. So basically, they're asking you how many there is during anaphase. Look, anaphase for humans is 92. It's not 46. If I actually had 46 in anaphase, you wouldn't end up with this. You would end up with 23 and 23, and this would have been a 23 and 23. So the cell will come out with 23 and 23. Is that really 23? No, humans are made of 46. So you have to start mitosis with the copy, which is 92 of them, right? 46 plus 46. So this is a scenario of a human 46, but this scenario is saying that this organism has 20 as a diploid. If it's 20, so it's 20 plus 20, so it must be 40. So the answer is C. C is the answer. Now let me just repeat because I know some people didn't get with the math. So one more time, make it more fast and quick, right? Okay. So here's the objective. You start, let's just say you start with 46 in the human. You want to go through the stages of prophase, right? Metaphase, anaphase, and telophase. And what do you want to end after cytokinesis? You want to end with a cell that is 46 and another cell that is 46. But you know that in anaphase, you are splitting it, right, into two. So half is going to one side and the other half is going to side. If you start with 46, here, what are you going to get? 23, 23. Is that right? No. If we remember from the cell cycle, we have G1, S, and G2. What happens in S? You had 46, you had to make a copy. So now what do you have in total? 92. So when you're going to start mitosis, you start with 92 if your diploid is a 46. In this case, the diploid is not a 46. The diploid is what? The diploid is made of 20. You want to end up with 20 and 20. So what do you start? With the double of it, which is 40. So when you get to anaphase right here, you're going to have 20 going to one side and 20 going from the other side. So once again, the answer is C. And that's pretty much. Thank you.